next session, the open floor session. So just a quick word about the open floor session. Some of you have already signed up for this session, but the floor will be open after the people who have signed up finish them. And you can, if you want to say a few words, you can come up and say. Depending upon the time, we would like to restrict it to 5 minutes or so, so everybody can have a chance to say what they want to say and share your experiences with David. So the first person that is going to come to the floor is David's daughter, Esther.
Nagraj, so thank you for everything, especially all the computer help for my dad. So my name's Esther, and Schlesinger Mida, I think one of the reasons I kept Schlesinger was because of my dad. A lot of pride. So I'm coming from a non-scientific perspective, so I just thought I'd summarize some of the most important and special traits about my father, which have made him great. And I was going to say, I'm sure some of the people in this room have experienced them, but you guys have mentioned a lot of the ones I'm going to say. I swear, Dad, I did this before I got here. So, this is Dr. David Schlesinger's top 10 secrets to having a fulfilling life and career, which I may or may not have learned successfully. First, pick a career with purpose that you enjoy and have a passion for. Second, make sure you have a patient, competent, and loyal spouse who is able to change all the light bulbs and handle any other household issues. Treat your colleagues like friends and your friends like family. Don't limit your passions. Be an expert on everything, even things out of your field. Always, always, always travel vigorously and everywhere and eat everything. Preferably with natives showing you around. And when possible, speak the language fluently and bring your kids. And never, never, never let anxiety stop you. Do everything anyway. And be early. And I mean be early for the time you're supposed to be early. And be humble, especially when your accomplishments speak for themselves. And pick the right people to mentor and see the best in people. And then keep your connections with the special people you meet. Like you're all here. And last but not least, keep working at twice the pace of the people half your age. And that's even if you're doing it unpaid as an emeritus employee.
Thank you, Esther. Our next invitee would be Dr. Igor David.
Thank you so much, Nagaraj, for organizing this and giving me 5 minutes to talk. I cannot speak about the science of David, even though I am not a non-scientist. In other words, I am or was perhaps a scientist, but we did never collaborate actually. And for a long time did not work in very closely related fields either, although just the last talk reminded me of ribosomes, which I used to be interested in as well a long, long time ago. But we knew each other maybe because when we started out and we were young, fields were small, so everybody in molecular biology knew everybody, and especially if you had an interest in sort of molecular genetics, developmental biology, and ribosomes, you would know each other. And so, after David and Alice came to Baltimore, we wound up meeting from time to time in a regular manner to enjoy together some of the other major interests in David's life, in addition to science. And of course, his daughter already mentioned some along these lines, and she knows him a lot better than I do, undoubtedly. But it is David's great interest in the arts, visual arts, music, theater, and of course that supreme combination of music and theater and opera. Also an interest in gardens, and as Esther mentioned, traveling. All of these things are something that David certainly enjoys a great deal. He must say himself, perhaps. I'm just concluding from observational evidence that he does. But I'm also really impressed of the really extensive knowledge that David and Alice have on all these subjects on arts, theater, gardening, and so forth. And one example of the sophistication of David's interest in music is his fondness for Haydn and Rossini. So leading to traveling to special festivals in Esterházy and Beethoven. And this is not sort of run-of-the-mill interest in music, but something more than that. And that's really all I wanted to say. I have been very happy to join in these excursions sometimes and doing these things together. And what I would like to do is wish David and Alice many, many, many more years to go to the museums and theaters and concerts and travels and have fun.
Thank you, Igor. I would like to call Dr. Miriam Garospi next to read one of the letters that we have received, to read that letter for our audience.
Thanks, Nagaraj. So this letter is by Laura Crisponi, and she wrote it in response to Nagaraj's invitation to join us today. She writes, "Dear David and Nagaraja, I regret to be not there with you to celebrate David's first 63 years in research. I had the privilege to meet David at the beginning of my career, and that was the opportunity of a lifetime, since I met a maestro, the wise that not only gave me knowledge but shared his being in every aspect of life, so wide opening my mind to a new way of working and thinking, and these things will always be. There is a flash that will always be stuck in my mind. During the relocation of the lab in St. Louis, David removed his jacket, got up on the bench, and helped us, students, researchers, technicians, to reorganize the shelves with the bottle solutions and other stuff. Something I never saw before or after. And there I realized what a real leader is. I'd have too many things to say, but you know I have never been good with words, so I prefer closing this email with this text from Tao Te Ching by Lao Tzu, chapter 22, titled Humility That Elevates. So this is the poem. Yield and overcome, bend and be straight, empty and be full, wear out and be new, have little and gain, have much and be confused. Therefore the wise embrace the one and set an example to all. Not putting on the display, they shine forth. Not justifying themselves, they are distinguished. Not boasting, they receive recognition. Not bragging, they never falter. They do not quarrel, so no one quarrels with them. Therefore the ancients say, yield and overcome. Is that an empty saying? Be truly whole, and all things will come to you. And Laura ends the message by writing, thanks for all, and although not physically, I'll be there anyway. Best wishes to all of you, and I'm going to try to read this in Italian. Un abbraccio forte e a presto. Love that.
Thank you, Dave. I next call upon Dr. Ilya Goldberg.
Thank you, Nagaraj, for organizing this and inviting me. As most of you probably know, I ran a research group in laboratory of genetics for a few years. What probably most of you don't know is I've known David and his family since before high school. My father, Greg Goldberg, did a postdoc in David's lab, and that's when I first met him. It was also in David's lab where I had my first lab experience. So this was, I think, the first year of undergrad. In the summer, I came to his lab. I wrote a little program to do sequence analysis, ribosomal DNA, of course. And eventually was allowed to wash some dishes and run some gels. Even I think the successful subcloning project. So the summer was quite full. I think that's a pretty good summer even these days. But in addition to that, David, of course, was concerned with my intellectual development and suggested I read some Russian literature, which was actually, it struck me because it was actually exactly the right thing I needed to read at that time. So quite insightful of many years of insights to follow. So after this experience, this was to me, it really changed my life because, it was sort of the typical lost undergrad, and this really focused what I wanted to do with my life, which is to do science. And it made me hunger for environments like this because, of course, as we all know, David surrounds himself with the best people, right? And that leads to the best environments. And I hungered for those and sought them out for the intervening years until I had the opportunity to come to the NIH and work in his lab. And this was, the other thing about David is that he's a great champion of technology and technology development. And I don't think he gets nearly enough credit for this, but I think that the people he assembled and the technologies he championed were a key factor in the Human Genome Project. And when I came here, I had the developed a bit of technology at MIT that had to do with large-scale imaging and microscopy. And of course, they would have the insight that the combination of technology, modern technology, and imaging can be quite powerful in assay development, etc. And so we worked on this for a while, but then quickly we realized that all the image analysis is wrong, and what we really need is a machine vision AI. We're going to build a laboratory of genetics. It seems prescient now, of course, to have done this, but this was close to 15 years ago, and back then, probably raised a few eyebrows. I know certainly when I talked about this back then, it raised a few eyebrows that we were going to do this, but we did because of the support and again, never-ending insights that come from David, and of course the people and the environment that allowed us to prosper. And of course, as soon as we built this, David said, well, really what you should do is look at medical images. And to me, this was very, very far from what I was interested in doing with this technology. But I did it anyway because, of course, by then, as we all know, you're better off following David's advice than not. And so I did. The group spent quite a few years looking at medical images, and now I'm co-founder of a startup in Seattle, Mindshare Medical, to apply this technology to do better diagnosis in various medical imaging devices. So I think that the championing of technology development and the assembly of great people were not only smart. Smart is never enough. They have to have passion, they have to be kind and generous. And that's a key thing that I always saw and tried to do myself in the people I surround myself with. So thank you for the great opportunities, David. And many will be years. Thank you.
Thank you. Next, I call upon Joseph Tran.
Okay, I want to thank everyone, and for Naira and very happy to give a chance to talk. So, very briefly, I came to the lab of genetics in 2003 as very, very naive, very technically competent trade and not really training. I was not very good at equipment. I was good at melting Agros gels, and there were some other items, spinning items that suffered a little bit in my hands. But the entire time I was here, David was incredibly patient, which kind of said, or saw that this person really likes to be kind, really enjoys the question, has a little bit to learn about doing it, and with a little bit of coasting along, has really helped me tremendously grow up to become competent and think about questions in a very scientific, rigorous manner. The other thing I would like to really say about my experience in lab genetics has been that David's incredibly wise. I had a project that was very difficult, perhaps beyond my technical skills at the time. David, you guided the RNase MRP project from an idea and from cloning to trying to make my into a paper. And it was something that I look at today extremely proud of. I mean, I contributed some things to it, but I mean, it was really like the organization, I never would have come up with it at that point in time. And I used that paper and I used that thought process as a model for how I think about science these days. So I've learned a lot from you about organizing things, the wisdom and leadership skill that largely adapted. The other thing, and I think this is really the biggest thing, and that's why I still to this day think about David as whenever it comes down to scientific questions. David, you believed in me. I think I didn't. There were times where I was shy, would not. Didn't believe in myself, but we were always happy that you can do it. And now I have continuing on the scientific journey with you, still believing in me and believing, I think, in all of us. And I really thank you for that. Everything that you've done scientifically for me personally. Thank you, David.
Thank you, Joe. I would now call upon Jun Ding to read a letter by one of David's best friend and colleague, Lucio Luzado.
This is a letter from Lucio Luzado. I first met David Schlesinger in February 1964 when he came to give a research seminar at Columbia in New York, where I had just started as a clinical research fellow under Dr. Paul Marks. David presented novel findings on the structure and function of E. coli ribosomes, that they were the workbench for protein synthesis had been only recently discovered. Given that my command of English was poor and my understanding of spoken American even worse, it helped a lot in our communication that David could also handle French and Latin. We promptly found out that we had been born in different continents, but within a week of each other. And I was enormously impressed that at the age of 27, David already had his own lab. This would have been unthinkable in Italy, and I later came to realize that it was not exactly the rule in the US either. Over the next 54 years, there has never been a time when we have been out of touch. To prove that, I have dozens of letters in David's fluently slanting left-hand handwriting. Even though that cherished physical record eventually had to yield to the irresistible email revolution. I think scientific continuity helped to create friendship. And once we were friends, I think we were seeking excuses to collaborate. The facts are that when, having moved to Nigeria in 1967, I was offered from elsewhere in the US a chance to do a short sabbatical. I came instead to David's lab at Washington University. And when my first PhD student was ready for his thesis defense, David came to the University of Ibadan as an external examiner. In brief, our friendship was steady and my admiration for David increased steadily. It is not for me on this occasion to list David's achievements, but what I have admired in him as a scientist has been his ability to sift through the details and then always focus accurately on the core question. His ability to use the most advanced technologies without ever losing sight of the biological objectives. His ability to cross disciplinary boundaries at a time when this was not at all customary. Indeed, in 1972, David and I were again together at the International Institute of Genetics and Biophysics in Naples, Italy, and almost immediately, David gave us evidence that certainly in his own case, microbiology might well have been the best background for making strides in human genetics. From then on, David's leadership in the sequencing of the X chromosome in the Human Genome Project as a whole, in the Sardinia Project, and in all the illuminating jewels it has generated have become part of history. David has helped to train innumerable young scientists from all continents, but particularly from the US and from Italy. They have learned from him the rigors of science and the passion for science, and they have felt his charisma through his bones. David's former mentor, James Watson, has been credited with the attitude that, contrary to what people think, including scientists' mothers, scientists are not necessarily nice people. It may not be wise for me to disagree with the Nobel laureate, but a prime evidence to the contrary is David. He was helped by his lovely family to always reconcile science with human relationships. On so many occasions, we have enjoyed David and Alice's hospitality. We have been privileged to know their daughters since they were babies. We have attended Esther's wedding, and Lillian has been with us in one of our most difficult moments. We are seeing with great affection David and Alice's children and grandchildren growing. What a beautiful family. Paula and I regret we are not physically there on this day of peace. We are certainly with you in spirit for this celebration of science and friendship. So this is the last sentence, and I'm going to try this in Italian. So, everyone for David and for all those who love you.
Thank you, Jim. Next, we have a brief presentation by Changi, one of our people working with David in Milan.
So with this chance, I also like to recall my time with David. So our time goes back to 20 years ago, in 1998. So October 1st, 1998, I started my postdoc training in the lab of genetics under David's supervision. And 2 days later, on a Saturday morning, David and I had a first meeting to discuss future research directions. And our meeting was in the Triad building, which was the home for the lab for about 10 years. I still clearly remember our first meeting clearly. In front of famous scientists, I was very nervous, and David was very kind, and he spoke not very slowly, but very, very slowly. Even so, I don't think I understood him completely, and I believe he didn't understand me either. So these photos show the Triad building. Inside of the building was like a hotel, and I believe many people still remember this building and remember the great time we had spent there. And fortunately, David and I could communicate more effectively as time went on. However, our communications were not always by the meetings, but by some unique ways. For example, through this mug or cup. So this cup is one of my favorites, and it was the first gift I ever received from David. And this cup was a memento of a genetic meeting held in Helsinki, Finland in 1996. And I believe Dr. Juha and Anand probably organized this meeting with David. So the drawings on the cup indeed outline my research direction. So as you can see, a gentleman trying to make a magic solution for his bald head. He eventually made one, and when he put on the solution to his head, he suddenly became a hairy person. So with this come, they suggested to me I should think this as my research direction, which meant understanding of the skin appendage development and the function. So in this direction, we could make some meaningful contributions, and we could co-author 28 peer-reviewed papers, and I was the first author in 18 of them. So it has been my privilege and a great honor to work with David for last 20 years, and I sincerely appreciate his long time support and teaching. And special appreciation to Alice Wei, Yulan, and I learned English from you for more than a half a year, which helped us a lot. And you also provided a lot of warm support. Thank you so much. And this last slide, let us recall some of our special time spent with David. The 2004 Christmas party. Even in the party, David was thinking seriously about science. And Nagaraja was very young in that time. And Weidong looks like a college student. And Minoru was always happy and optimistic. So this is NIA Picnic Day 2007. Someone looks like me. So this picture Minoru showed, it's David's birthday and the 10th anniversary of the lab of genetics. Halloween 2008. And the Christmas party 2009. And September 20th, 2011, David's birthday. And the Christmas party last year. And they were the leaders of the lab of genetics. And congratulations, David, and the best wishes on your new chapter in life. Thank you.
I would like to call upon Dr. Buddy Branson as next presenter.
Thank you, Dr. Raj. It's a pleasure to be here with all of you, as well as the pleasure of having collaborated with David for years. And I'm happy to take part in the celebration of his scientific career. This is probably a midpoint. I doubt that it's over, and I think that what he's done is provided a base for where it's going to go next. So, you all know the story. David left St. Louis and came down here. I stayed in St. Louis and thought, boy, oh boy, what's going to happen? And once you get addicted to being part of a very large project, it's really hard not to continue in that sort of vein. So I joined what's known as the Blue Green. Which was an idea that was part of the National Institute of General Medical Sciences to put together a bunch of investigators of varying kinds of backgrounds and expertise to focus on the problem of inflammation and the host response to inflammation. And there are many subsidiary diseases that result from that are very significant to solve. And as part of my activities in the Blue Grant, I got interested in systems biology. And we wanted to use that as an approach to see if we could put things together, understand what goes on in inflammation, and do something about it. And the problem that you face, and many of you I know already know this, is that you have many reactions in different parts of cells, in different cells altogether, and in different organs of the body. So how do you put that all together? So that's why the systems biology approach seemed like a reasonable way to go. On the road, we looked at the human response to endotoxin, lipopolysaccharide. And the reason for that is we weren't sure if the methods that we were using at that time were going to work. Are we going to see things at the molecular level that we expect? And can you really measure them accurately? So we subjected some volunteers, human volunteers, to endotoxin under controlled conditions, of course, and took blood samples at various times and asked what happens to the white blood cells. And we were actually able to separate the white blood cells into the major monocytes and lymphocytes, etc. So, I'm going to show you a couple of results that we got here that go back to whether or not the systems biology approach is telling us anything. And then that was quite a few years ago that we did that experiment, but every now and then I go back to it and there's still things to analyze from it. It's amazing what comes out of it. Currently I'm working on problems with coagulopathy, but they also are subject to these kinds of approaches. Yeah, so at 0 time, this is what the white blood cells look like. And then at 2 hours, you can see the associations that are formed. And everybody's had experience now making these little star charts and looking at the association and the things that are happening. But this is taking a much more general approach. What's happening at the cell membrane? What's happening at the nuclear membrane? What's happening inside the nucleus? And then the idea is to get a feeling for this that's changing. When I read papers that involve these kinds of approaches, what we have are snapshots of what happens at various time points. It does not give you a feeling for what's happening during time, while those cells are growing, aging, and eventually differentiating into what they're going to do. So, well, okay, so the slides didn't work quite the way I thought they would. While this was going on, I became a referee at USA Cycling and helped run bicycle races. And this was done both on the national and international national scale. So what I have here, you're not going to see unfortunately, is a picture, again, a snapshot of what happens during a bike race. And so you have a lead group, then that maybe a mile behind the lead group is the main pack. The main pack could easily have 100, 150 riders. And if you've ever tuned into the Tour de France on TV, you've seen part of this. Once again, you get a snapshot of what happens at one point. And I took a picture of the riders coming up a very nasty hill with about a 10% grade, and they're huffing and puffing. And the interesting thing is that at that point, you don't know who the winner's going to be, because you're looking at a snapshot of what's going on. You're not looking at the whole point of progress. So, it also reminded me very much of what we're trying to do in our research, and that is learn what happens during the progression of the metabolic and genetic events that are occurring in the cell. So that's what we're actually trying to do, and unfortunately I see that missing in an awful lot of the papers that I read. There you are. And so you're going to ask, where do we find David in that bike race? Well, you don't. We never got David interested much in cycling. And I'm not going to tell you where I am, although if you looked at the end, you'd probably get a good idea. I mean, like an hour behind the riders. But you kind of get a feeling for what's going on. If you could shrink yourself down to the molecular level and looked at what was going on in the activities of the cell, this is what you would see at a given point in time. You would not know who the winner is going to be. And it's amazing how the dynamics change during the race. So David is responsible for all that. When we came up with the idea for actually attacking the notion of the Human Genome Project, can we actually make a physical map of the human genome? Unbelievable cries of negativism. But, the ones that thought, oh, what a great idea, we need to do that. So when David invited me to come out of cold Chicago and come down to warm St. Louis, I thought, yeah, this is definitely the way to go. So one of the things I learned from David, I know we've heard some great things about what everybody's learned during the course of their work. Yeah, so I have a couple of things up here. Look, I'd like to start with the ones at the bottom. I still remember when David said, the less you know of something, the more you can make of it. And he was kind of waving his arms while he was saying that. And I thought, yeah, that's really true. And that has come up a number of times. Other things that people worry about is stealing your data. What does it mean if they're going to steal your data? And as he points out, your value in science is negligible if they don't want your data. So let them go ahead and steal it. So then David had an effect on other important people on the planet. One of them is Daniel Kahneman. And he formalized one of the things, and that is not knowing much makes it easier to fit everything you know into a coherent pattern. So I'm sure he's thanked David for that, having stolen the idea, of course. So, I want to thank Nagaraj and his colleagues for organizing this symposium and for getting to rub shoulders again with people who I've known at various times. And I guess we will continue with more of the celebration for David.
Next, I'd like to invite Dr. Weilong Wong to read a letter that was sent by Dr. Tony Angelos.
Okay. Dear David, you may not remember the first time we met. It was during a hot summer day when you visited the ancient underground Roman ruins in Fano, my hometown. It was a lovely day which we spent enjoying the glory of civilization long gone while talking about life, science, and cracking jokes. When you did, I was too intimidated to do so. I still remember that day as a humbling and inspiring experience, the beginning of a new and exciting journey. Coming from a PhD heavily based on proteomics with a little background in genetics, I'm grateful you took me the chance and allowed me to join the lab. I had the greatest of experience at LG, and some of my fondest memories bring me back to that third floor of the trial building on Ka-Xiao Drive, and later the 10th floor of the BRC. I miss our brainstorming sessions in your office where we would discuss the best ways to answer some scientific questions on how to troubleshoot a problem, or just talk about current events, which often ended up in laughing about the absurdities of Italian politics. I miss our conversations during coffee breaks when I would come to your office to get that precious Italian espresso grind you kept stashed in your desk drawer. That coffee we would brew on a hot stirring plate. It seems we didn't make difficult experiments only inside the lab after all. Thank you for always being so positive and supportive. You have been the most wonderful mentor I could have ever hoped for. I still have to find another person who would walk along the lab benches, smile, wink, and raise a fist as a gesture of encouragement. Thanks for that. There were days when you made a difference. Speaking of scientific achievements, John of Salisbury said, "We are like dwarfs perched on the shoulders of giants." And thinking of you, I couldn't agree more. Thank you for being such a giant, carrying so many of us on your shoulders. Hello.
Thank you, Adam. Next, I would like to run a slideshow that was sent to me by Joe Porta, one of the postdocs that worked with David during his tenure in Washington University, St. Louis. You see Joe Porta there, and I'll just advance the slides so you can read through as we go. So that kind of more or less concludes, but we have a couple more steps to go through. And now the floor is open for anybody else who want to say a few words before I call the meeting. You will each have maybe 3 minutes or so.
Thank you, Nagraj, for organizing this. I first declined to say anything. I had a little social phobia, and I don't like to get in front of people. But when I saw David, I felt like I had to say something. I graduated from college in 1989 with a molecular biology degree. Molecular biology was kind of new back then, and I loved it. And wasn't sure what I wanted to do. My home is in St. Louis, and didn't have the grades to get into medical school at that time. I was fortunate enough to get a job as a laboratory tech in David's lab. And I got to know David over the 2 years that I was there. David is the complete opposite of my father. He is a cerebral, gentle, and humble man. My father, who's also very kind, was the Cro-Magnon man. He was a very tough man. He grew up post-Korean War, and he used to say that if you didn't know how to fight, that you would get your lunch money and books stolen every day. So my father was the Korean national judo champion. He was in the special forces of the ROK, or the Republic of Korea, South Korean Army, which is a very tough military. And he used to say to me, Jay, human beings can be the most beautiful animals in the world, but they can also be the most dangerous animals. Be careful, because people will betray you, they will be deceitful, they will be ungrateful, they will underappreciate you. That was my dad. And the older that I got, I was younger, I was like, oh, whatever, Dad. And, but the older that I got, I realized that some of that is true. Life is not easy, and it can be ruthless, especially in science and medicine and some competitive fields. It can be hard. But getting back to David, I want to say that, I've had many teachers. I did, when I was at Princeton, I had a thesis mentor. I had many attendings in medicine, and many chairmen that I had to report to when I was in academics. And many times they were interested in how much money I could bring into the department, how many book chapters could I write for them, et cetera, et cetera. With David, when I worked in his laboratory, he took me under his wing, and he was interested in me as a person and in my happiness. He helped me get into an MD PhD program, which opened many doors for me. I am working as a physician in a niche that I absolutely love. I am privileged and fulfilled in taking care of difficult skin cancer patients, and that wouldn't be possible, I don't think, without David having helped me. I want to thank David for my career, for being so generous to me. He's the most generous mentor. And remember, I was just a laboratory tech, and not a very good one at that. But some of these real men that were supposed to be my real mentors, David has been more of a mentor to me than any of those people. And I want to thank him for that generosity. I want to thank him for inspiring me to be a better mentor myself and to be a better human being. And I want, so it's obvious how extraordinary and brilliant his scientific career has been. But I think what is more extraordinary is the way he has lived his life. I've come to realize that success in life is not how much money that you accumulate, how much stuff that you accrue, maybe not even how many papers that you publish, but it is about the relationships that you have. It is about your life experience. And David's, all the love in this room and beyond this room, and all the people that he has touched and helped, is a testament to his success in that way. I want to thank him for being the surrogate father figure that complemented my dad. I want to thank him for making me realize that human beings can be the most beautiful animals in the world. Thank you very much, David. Very much.
Next, Dr. Ben Kwan is here.
Well, when I was a medical student at WashU, I spent a summer in David's lab. That's about it. But what I want to say is, last year when I went back to WashU for my 50th anniversary reunion, I was surprised that during the banquet for the reunion, David and Alice was there, and David was honored with the award of Distinguished Professor for Washington University. Since it happened last year, so most of you don't know about that, but this is a very honorable award by WashU to David. That's it.
Thank you. John?
I was recruited by David in 2011 as a staff scientist to the lab. Since then, I have been closely working with David in Sardinia project and in various other projects. This has been a wonderful scientific journey for me. And in this journey, David's passion for science impresses me the most. He is always curious about the unknown. He's always thinking about new ideas, and he's always willing to take the risk to challenge the difficult problems. I think it inspires all of us who work closely with him. It inspires us to try to explore the unknown and also try to always make new discoveries in our scientific life. So for that, I want to thank you, David.
Thank you, Jim. Next, I call upon Ji Zhang.
I would like to after this he was saying that David was support and help in my career. So I came to DLD Dr. Wick once again, 2001, and I was not direct member of the David's group, but during the 17 years, David gave me a lot of advice and taught me many scientific research and in many ways. So I don't know how to express that my appreciation for David's support and help, as well as Alice for wonderful English, teaching my English, and but this is my great fortune and experience in my life. I thank you, David and Alice.
Thank you, Ji Zhang. If nobody else wants to take the floor, I think we can proceed to the next steps. I have a few words to say to David before I introduce him to come to the podium to give his presentation. So I first met David in the summer of 1989 when he was visiting for a genome meeting in Bethesda. I was exploring an opportunity for my second postdoc, and I had established the contact with David through Buddy Bronstein, with whom I worked at the University of Chicago. Even though my publication record was not so great, I think he had already saw my CV and saw that I had graduated from the University of Chicago. So I think he had already decided that he could take me into his lab. So I think when we met, he suggested that we have some coffee and chat about some of the work that they were doing. And he was highly enthusiastic about the genome mapping project that the project had just started. He was running the center in Washington University and suggested that it would be a great opportunity for anyone who is interested. While we were talking, I realized that it is not unusual for me that I didn't have any money in my pocket to pay for the coffee that we were going to have. So I told David that I had to come clean and tell him that I didn't have any cash to pay for the coffee. His immediate reaction was that how in the world would I get home if I ran out of my gas in the car? It is this extended implication of one not having money on hand and how one would deal with an emergency was more concerning to David than the current problems at hand. That is David in short, always thinking about the welfare of others. Of course, I joined David's lab in St. Louis the following year. So this was my second postdoc stint, and it has been the best career decision that I have ever made in my life. David has always empathized with his associates' situations and worries about their future. He has enriched our lives with his in-depth knowledge of music, art, and introduction to Missouri and Maryland local wines. Through his memorable field trips to wineries on many weekends, these trips and experience will always live in our hearts forever. His travelogues are full of details and virtually take you to the places where he has visited and make you visit them as well. His life values have extended to our children. He has been part of our family. He attended our kids' award ceremonies, graduations, and weddings, and music festivals that our kids participated in. I want to thank him for making us a part of his family. We come to know Alice, Lily, Esther, and their children and their proud achievements. And his scientific ideas might seem at some point to a novice are off the wall. But pursue them, you'll find that they are very well thought out, researched, and with a plan of action. It is an understatement to state that working with David has been a privilege and good fortune. You have taught me how to write scientific papers, contributed to improving my English language. I noticed recently that there is less and less of reading when I submit drafts for revisions. Thank you for the opportunity to be associated with you during these 27 years of my scientific career. Hopefully now that you can think less of science and its pressures and dive into and pursue many of your other interests with equal penchant for you have for science. Thank you, David. David graduated with chemistry and English majors from University of Chicago in 1957 and got his PhD in biochemistry in 1960 from Jim Watson's lab at Harvard University. Finished his postdoctoral training with Francois Gros at Pasteur Institute and joined Washington University School of Medicine in St. Louis. At Washington University, he served as professor in the Department of Molecular Microbiology, professor of microbiology in medicine, and professor of genetics between 1972 and 1997. He was head of the Genome Center throughout its existence at Washington University, leading to a successful construction of a sequence tag site-based physical map of the human X chromosome. And David joined NIA in 1997. David has earned numerous awards during his career. One of the earliest was the prestigious Eli Lilly Award given in 1969. You heard about it before. He has received honorary MD degrees from the University of Uppsala in 1994 and from the University of Cagliari in Italy in 2002. He is also an honorary citizen of Lanusei in Sardinia, Italy, as was announced by Francesco. He was recognized with NI's Distinguished Investigator Award in 2011. In 2017, he was given Distinguished Service Award from Washington University in St. Louis, Missouri. He has been invited to give lectures internationally. One of the most recent series was Sekiguchi lectures at Keio University in Tokyo, Japan, that Minoru actually alluded to earlier. He has served on many journal editorial boards, ran X chromosome workshops for several years. He is a member of scientific international organizations. He has led the longitudinal studies of aging-associated conditions in certain population cohort following his arrival at the NIH, which has led to many landmark publications and collaborations. His publication records now sum up to close to 580. We now welcome David as a scientist emeritus into the National Institutes on Aging. And he keeps writing more papers, of course. I now ask David to give his elaboration of the scientific achievements in his own words.